Hi everyone, welcome to IGCSE Study Room where you can revise biology topics from the Cambridge IGCSE syllabus. This video summarizes Topic 4 Biological Molecules. The bodies of all living things are made of many kinds of chemicals. The three main categories of molecules in living organisms are carbohydrates, fats, and proteins. Since these molecules contain carbon, they are known as organic molecules. These are the chemical elements present in these molecules. So as you can see, carbohydrates, fats and proteins, all three contain carbon, hydrogen and oxygen. But only protein has nitrogen additionally. And proteins even contain a small amount of sulfur. Let's look into these chemical elements in more detail. We need to know that large molecules are made up of smaller molecules. The first is carbohydrates. They are large molecules made up of simple sugar molecules. The simplest form of sugar molecules are also known as monosaccharides. Glucose is an example of a monosaccharide. When two glucose molecules join together, maltose is formed, which is a disaccharide. When lots of glucose molecules join together, starch, glycogen, or cellulose can form. They are called polysaccharides. This picture illustrates the three types of carbohydrates. A monosaccharide is a single sugar molecule. A disaccharide is when two sugar molecules are linked. A polysaccharide is when many sugar molecules are linked. Examples of many glucose molecules joined together are starch, glycogen, or cellulose. The next category of biological molecules are fats or lipids. Fats are large molecules made from smaller unit of fatty acids and glycerol. Their basic unit is one glycerol molecule chemically bonded to three fatty acid chains. Fats that are liquid at room temperature are called oils. So this picture shows that each fat molecule consists of one glycerol molecule bonded to three fatty acid chains. Next is proteins. Protein molecules are made of long chains of smaller molecules joined end to end. These smaller molecules are called amino acids. There are about 20 different amino acids. The amino acids can be arranged in any order, resulting in hundreds of thousands of different proteins. So, different combination of these amino acids will give rise to different proteins. Even a small difference in the order of amino acid results in a different protein being formed. Here is an illustration of amino acids that when combined forms protein. Next we will be looking into the food tests. Different tests can be performed on food to identify if they contain starch, reducing sugars, proteins, fats and oils, or vitamin C. Let's first take a look at the test for starch. In order to find out if some food contains starch, we should add drops of iodine solution to the food sample. If the food sample contains starch, the color will change from orange-brown to blue-black. Here is a picture to show this test. As you can see, a few drops of iodine are poured onto a potato. Since potatoes contain starch, the orange-brown color of the iodine turns blue-black. The next food test is for reducing sugars. We must first add Benedict solution into the food sample solution in the test tube. Heat at 60 to 70 degrees Celsius in a water bath for 5 minutes. Take the test tube out of the water bath and observe the color. If reducing sugars are present in the food sample, the solution will turn from blue to orange or brick red. This diagram shows the food test for reducing sugars. Benedict solution is added to the food sample, which is heated in a water bath, and if the color changes from blue to orange, it confirms a positive test for reducing sugars. The next food test is for proteins. Add drops of biuret solution to the food sample. A positive test will show a color change from blue to violet or purple. This illustration shows what happens when a food sample is tested for protein. Since the color changed from blue to purple, it means that the food sample contains protein. Now the test for fats and oils. The food sample is mixed with 2 cubic centimeters of ethanol and shaken. The ethanol is added to an equal volume of cold distilled water. A positive test will show a cloudy or milky emulsion forming. So, if we want to check if a food sample contains fats or oils, we must mix the food sample with ethanol, and then we must add that to some cold water. If the food sample contains fats and oils, a white cloudy emulsion will form. The final test we'll be learning about is the test for vitamin C. Add 1 cubic centimeters of DCPIP solution to a test tube. DCPIP is blue in color. Add a small amount of food sample in the solution. A positive test will show the blue color of the dye disappearing. This picture shows that when the blue DCPIP solution is mixed with the food sample, the blue color turns colorless and disappears. This confirms that the food sample contains vitamin C. Finally, we'll be learning about the structure of a DNA molecule. DNA or deoxyribonucleic acid is the molecule that contains the instructions for growth and development of all organisms. It consists of 2 strands of DNA wound around each other in what is called a double helix. Each strand contains chemicals called bases. Hydrogen bond between pairs of these bases hold the strands together. These bases always pair up in the same way: A with T, or adenine with thymine, and C with G, that is cytosine with guanine. Full names are not required to be known for the exams, just the letters. But you must know which base bonds with which. Here is a picture to better understand the structure of a DNA molecule. You may notice a double-stranded helix held together by base pairs. The base pairs are held together by hydrogen bonds. The base pair always pair up in a particular way. A pairs with T and C pairs with G. So that sums up the main things to be learned in Chapter 4, Biological Molecules. Hope you found it useful. Thank you for watching, and please don't forget to subscribe to IGCSE Study Room for more biology revision videos.